Hello, everyone, and welcome to today's webinar, Cell-Free Tools to Revolutionize the DNA Construction Paradigm. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Oricero Genomics. To learn more, visit oricero.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. In addition to submitting questions, we request that you participate by answering poll questions during the webinar when prompted by our speakers. To submit your answer, click on the choice that best represents your answer. I'd like to now welcome our speakers, Shige Sasaki, COO, CBO, Oricero Genomics, and Kazu Isono, Global Marketing Manager, Oricero Genomics. Shige and Kazu, you may now begin your presentation.
Thank you, Mary. Okay, let's get started. Okay, at first, I'd like to introduce the Oricero Genomics as a company. So we are developing the unique cell-free plasmid DNA engineering and amplification technology. The company technology is covered by 6 patent families in the major market. And currently, we have launched 2 of DNA synthesis tools in the global market. You can buy it from everywhere. And also, we are conducting the custom service, which can construct and amplify up to 200 kb size of plasmid DNA. Okay, let's talk about current DNA cloning technology, which is cell-based cloning. As everybody knows, this is the E. coli system. But this cloning process has gone largely unchanged for nearly 50 years. But as you know, the industry still rely heavily on the PCR and this cell-based cloning system. Both are decade-old methodologies and are deeply rooted in effectively in all aspects of biological research and drug development. However, they are not without their limitation and drawback. Okay, this slide shows the key challenges on the cell-based cloning. I think you may facing every day. So high GCR80 contents and repeat sequence cannot be amplified in the E. coli system, and it's time-consuming and multi-steps and multi-day process is required. And cytotoxic sequence is never working. And also it's low throughput and low diversity and difficult to automate it. And for manufacturing, cell banking is required, which is time-consuming and a huge footprint of facility is required. So good example is that the messenger RNA vaccine, as well as therapeutic antibody for COVID-19, is increasing reliance on the genetic material as their engineering and technologies. Now, I would like to introduce Oricero cell-free plasmid DNA technologies. Oricero successfully fully reconstituted into a single reaction based on more than 20 proteins, which are essential for E. coli genome propagation. It has accurate cell-free assembly and amplification ability of circular DNA and solve the problem of PCR and cell-based cloning. So there are 3 key features here. So first one is rapid, because just a few hours enzymatic reaction in the one tube. And the second, it's unlimited. It is applicable to any complex DNA and scalable to gram scale. Efficient, the carry out in the small facility, no need to large facility and labors. Okay, before I move to the more detailed slides, I'd like to have a poll question here. So please take a time to make an answer. So what are you struggling with E. coli-based cloning now? Okay, here we go. Let me check the answer here. Oh, still people is working, I think. Oh, that's interesting. Okay, now I can see that topo is a GC reach 80 reach, such kind of difficult sequence, and followed by large DNA construct. That's a very interesting result. Okay. Okay, thank you for your answering. Then, okay, now I pass the ball to the Kazu to talk about more detail about technology and data about our technologies.
From here, I explain our technology. First step is preparing the DNA fragment to be cloned. This DNA fragment can be PCR product, restriction enzyme cut, or synthesized DNA. These and ORC sequence are joined together to form the circular DNA. ORC is the replication origin of the E. coli chromosome. Therefore, the circular DNA containing ORC become the E. coli genome-like structure, and the protein involved in the replication of the E. coli genome enable this circular DNA to be replicated and amplified. Circular DNA design is simply place the overlap sequence at junction of the adjacent DNAs. Design detail is explained in later slide. By simply mix all the DNA fragments that compose the circular DNA with the assembly reaction solution and incubate at this isothermal condition for 30 minutes. It complete the circular DNA containing the ORC sequence. Then, this circular DNA solution mixed with the protein cocktail which involved in the replication of E. coli genome and incubate also at the isothermal temperature for 2 to 6 hours. Only the circular DNA will be selectively amplified, and this amplification product can be used in downstream application with minimum purification, such as transfection. In this slide, it shows example of amplification. In the 8 kb and 50 kb amplification example with ORC, it can be seen that each circular DNA is amplified. In addition, amplification product becomes supercoiled DNA. If you want to test the exact length, it's recommended to treat the amplification product with appropriate restriction enzyme to make it linear and analyze it by electrophoresis. This slide shows the amplification result of base biased sequence. The left side is the result of cell-free amplification of the 9.3 kb plasmid with 70% GC content. You can see that only the circular DNA containing ORC is selectively amplified. On the right is the result of the cell-free amplification of 7 kb plasmid, which has approximately 80% of the AT content. You can see the supercoiled DNA is also amplified here. In the lane 3, the nicked plasmid and incorporated amplification products are digested by the small amount of exonuclease. Only supercoiled DNA remain. In this way, even if the sequence is generally difficult to amplify, our cell-free cloning technology can reduce the sequence selectivity. In this slide, I will explain the assembly reaction. Here is an example of binding 10 DNA fragments with 600 base pair lengths. Adjacent fragments, such as DNA fragment 1 and 2, have homologous sequence as 40 base pair at the junction. Similarly, designed to have an overlap sequence at the end like 2 and 3, 3 and 4. Design is similar to the Gibson assembly, but overlap arrangement have different lengths. In the assembly reaction, enzyme digests single strand from the 3' end of each DNA fragment to make a 5' protruding. And the complementary base anneal. At this time, our technique promotes the binding by enzymatic annealing instead of thermochemical annealing like PCR primer. Therefore, the reaction time of the assembly was shortened to 30 minutes. On the right, it's an electrophoretic image of the assembly product. The bound DNA fragment appear in ladder pattern, and you can finally observe up to the desired 10 bounds. Here is an example of connecting 50 DNA fragments and 1 ODC to construct a circular DNA of 27 kb. The overlap between the adjacent DNA was designed with 60 base pair. After assembly reaction, the product showed the ladder appearance of several DNA fragments bound together. Also, it cannot be seen here, 51 DNAs are finally assembled. A part of this product was mixed with an amplification reagent and amplified. When the amplified product was checked by electrophoresis, the target 27 kb supercoiled DNA was confirmed. The assembled product were used for the amplification reaction, but it also contained non-circular DNAs. But it doesn't matter. The template requirement in the principle of amplification were two. One is containing an ODC sequence, and the other is circular DNA. So, even if it's a fragment containing ODC, it will not be amplified if it's not circular. It's also a feature of our technology that we can selectively amplify only the full-length construct without purifying the assembled product. Here we show the possibility of application. Upper one is an example of AAV application. As you know, AAV require multiple plasmids, but you can cut out the gene regions that have the function you need. Then integrated them into one vector. Since E. coli is not used, the antibiotics resistance gene had removed to create an integrated construct. Following is an example of entire genome cloning. An adenovirus genome has 35 kb linear DNA. So it converted into circular DNA with an ORC sequence. Finally, bottom one is an example of amplifying 140 kb of a baculovirus. By inserting the ORC sequence into the circular DNA, we could amplify 140 kb of baculovirus genome. In this way, our technology has potential to be applied to various applications by allowing the length of the clonable DNA than conventional technology. This slide is a hot topic. This data is an example of cloning the entire SARS-CoV-2 genome in collaboration with Kyoto University in Japan. Since SARS-CoV-2 is a single-strand RNA genome, it was designed by dividing it into 6 fragments with 5 kb cDNAs. In other words, this construct is consisting 7 fragments which of 6 cDNA and 1 ORC. In this way, the entire genome was amplified cell-free 30 kb and transfected into culture cell for genome analysis. Practical applications like this have already begun. In general, AT-rich sequences works against amplification. But with our cell-free amplification technology, the effect is relatively small. We incorporate the synthesized 160 base pair of poly-A into 4.3 kb circular DNA with ORC. Then we compare the yield. The cell-free amplified product was analyzed by cutting the backbone of circular DNA by restriction enzymes and electrophoresing them as 2 linear DNAs. We can see the longer DNA fragment is common size, but the shorter DNA fragment is not. The poly-A fragment shows a higher molecular weight at the 160 base pair greater. Thus, it was shown that even if the sequence has a poly-A sequence, our technology does not affect on amplification. In this slide, I mentioned yield. With the conventional large-scale culture method by E. coli, it took time and effort. The most complicated task is to generate the plasmid DNA from cultured E. coli. However, with our technology, plasmid preparation can be completed on the benchtop in short time. Present our reagent generate 1 microgram cell-free amplified DNA in 10 microliter reaction. But what should we do if higher yield are needed? In-house experiments, we have found that there is a linear correlation between the reaction size and yield. In other words, 1 microliter reaction yields 0.1 milligram, and 1000 milliliter reaction yields 100 milligram of amplified product. Moreover, the reaction time is not much different from present our kit. I can suggest you the large-scale plasmid prep is possible to reduce the time drastically. Of course, There is no need for large culture tank, so capital investment can be minimized. In 2023, we plan to start shipping this large-scale cell-free amplification reagent as GMP grade. This slide shows a technical comparison for amplifying DNA. The main DNA amplification techniques are PCR and E. coli cloning. But there are some disadvantages at each technique. For example, in PCR, it's the amplification length and fidelity of the product. Also in E. coli, it takes the time to culture. Our technology is new technology that solves these problems. Cell-free amplification of DNA up to 50 kb in several hours. Fidelity is 100-fold greater than PCR's one. Also, any difficult sequence are applicable. So we can say it's just the beginning of the new generation of the cloning technology. From here, we will explain about our current product. Two kits are currently available. The first is the Oricero cell-free cloning system. The kit that creates an amplified circular DNA with novel Oric construct. This kit contains 5 reactions of assembly reagent and 10 reactions of amplification reagent. Second is the Oricero cell-free switching system. The kits that allow you to introduce Oric into your existing plasmid for cell-free amplification. This kit contains reagent for 5 reactions. Finally, the Oric amp kit contains only reagent for amplifying circular DNA with Oric. This reagent is commonly included in the Oricero cell-free cloning system and the Oricero cell-free switching system. I introduce the outline of the product of the switching system and the amp kit from next slide. The explanation so far has been about newly creating and amplifying the circular DNA using the Oricero cell-free cloning system. From now on, I would like to explain another reagent, Oricero cell-free switching system. This kit contains a DNA fragment called the SS Oric cassette. This SS Oric cassette has a homologous sequence on commercially available vector at the end of the Oric sequence. This homologous sequence can be used to introduce an SS Oric cassette into the existing plasmid vector by homologous recombination. After introduction, it can be used as a template for cell-free amplification. However, using the Oricero cell-free switching system does not allow the Oric introduction into all vectors. Some of the popular plasmids described in the table can be introduced because they have a common sequence in their backbone. In case there is no homologous sequence on your vector, you can introduce Oric with Oricero cell-free cloning system. This homologous sequence is described in the user manual. So first check whether the sequence is included in your vector before using it. In addition, even if the homologous sequence overlaps with gene region on, the sequence does not change due to homologous recombination, and this gene is not disrupted. Here, the data in which the SS ORC cassette is inserted is shown. Once inserting the SS ORC cassette, total length elongates the 426 base pair which corresponding to the ORC sequence. In addition, this electrophoresis diagram shows that ORC integrated DNA show the stable yield as equal to or higher than the non-inserted circular DNA of E. coli production. The ORC amp kit is for cell-free amplification kit. It does not contain assembled reagent. This kit can re-amplify the already created construct which containing the ORC. Therefore, it can maintain the construct in true. This eliminates the need to store E. coli in glycerol stock anymore, and the tiny amount of ORC contained the construct can be amplified in half days at any time. Of course, since E. coli is not used, purification of the amplified product is easy. The reagent master mix included in this kit has optimized the enzyme and the substrate on standard protocol. Therefore, if all substrate in this master mix is used, concentration will be 100 nanogram per microliter and 1 microgram yield. Present reagent kits are capable of cell-free amplification of circular DNA up to 50 kb, and the yield is 10 microgram per kit. Our kits have already been shipped more than 700 kits since product launch, and the purpose of them can be roughly divided into four. They are 2 coronavirus, 4 long DNA clone, 4 difficult clone which does not grow well in E. coli, and process enhancement. We are proud that our product have answered to the customer's needs. So, what are the unmet needs? It can hear the voice of customers who need longer and larger amount of DNA. These are not satisfied with the specification of the current kit. To meet their need, we can offer custom service. In fact, our technology is possible to create the construct up to about 200 kb, and it has been verified by in-house experiments. Larger amount of the yield can be obtained by using the specialized enzyme cocktail from commercially available kit. Therefore, if you need more length or yield longer than current kit, please use this custom service. Orishiro Genomics is a Japanese company. Headquartered is in Tokyo. Also, we have a U.S. office in Massachusetts. In addition, We have several contributors in Europe and Asia, and we have expanding our sales area all over the world. For product inquiry, please contact us via our website or email here. We will develop and launch the new product continuously. Thank you very much for attention.
Thank you, Shige and Kazu, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how short or long of DNA can be amplified?
Okay, so the short one is, so our system can be applicable from the 200 pp, and the large size, so that you can amplify up to 50 kb with the cloning system, and up to 200 kb with in our custom service.
All right, thank you. Next question. What are the major differences between Ori-Zero assembly and Gibson assembly?
Yeah, I answered from my side. So the Gibson assembly and the differentiation of the Gibson assembly and our assembly technology is, so the Gibson assembly is just the thermal annealing, but so that our assembly technique is so the it's called the enzymatic annealing. So on the process of the annealing, so that in our technology, so the some of the special enzyme assist to anneal to increase the efficiency.
All right, thank you. Next question. Can the amplified DNA be used for transformation of E. coli and transfection of cultured cells?
Richards.
You
Okay. Well,
let me read the question one more time.
Yep.
Can the amplified DNA be used for transformation of E. coli and transfection of cultured cells?
Yeah.
Yeah. So yes. Go ahead, Kazu.
Yeah, it's possible to use that everything. So the amplified DNA is just it's almost equivalent of the regular circular DNA. So it's possible. Yeah.
All right. Next question. What should I pay attention to when designing an assembly reaction?
Yep. So the assembly reaction. So I will explain the some of the rule. So the so design it's required overlap length. It's 40 to 60 base pair. It's required. So the shorter, longer, it's not especially the shorter lengths. So for example, the 6 or 8 base pair overlap, it's not applicable. So the required 40 to 60 base pair. It's required. It's the attention point 1. And the second one is so the total amount. So you have to use a few picogram to up to the 20 nanogram. So the amount range should be within a few picogram to 20 nanogram. And the third one is so the mixing recipe is very important. So I mean so the equal molar rate is very important. For example, the OEC and DNA fragment should be cocktailed 1:1 ratio. And this one, this is the last one. So to approximate the DNA length of the fragment. So, for example, the 10 kb DNA construct. So, the fragment to use the two fragment, you should so the 5 kb and 5 kb two fragment. It's much better design, and so the 3 kb and the 7 kb, or it's not good design. So the approximate the DNA length. It's very important. These 4 points it's a good rule of the DNA design.
All right. Thank you. Next question. What should I pay attention to when inserting the OREC fragment?
Yeah. So OREC, inserting the OREC fragment, just position as it's possible to insert the OREC fragment anywhere. And so there is no restriction on the insertion direction. So.
All right. Thank you. Next question. What is the error rate for the isothermal amplification step?
Error rate is much greater than the PCL. Roughly, the PCL is about the so the our amplification our fidelity is about the 100-fold greater than PCL.
All right. And our next question, what's the I'm sorry, excuse me. We are running out of time for questions. I forgot that we have one more poll question that we would like our audience to answer. If we could pull up that poll question. What are you interested in Oriciro's cell-free cloning technology? Transfection efficiency, DNA cloning difficult by E. coli, large plasmid DNA construct, poly A isn't lost, library preparation for MABs. Remember to select your answer, click on the option that is the best choice for you. In our poll question, what are you interested in Oriciro's cell-free cloning technology, transfection efficiency, DNA cloning difficult by E. coli, large plasmid DNA construct, poly A isn't lost, library preparation for MABs. You have a few more seconds to make your selection, and again, click on the best choice for your answer. Thank you, Shige and Kazu. Do you have any final comments for our audience?
Thank you very much for today's attention. We really are happy to have more questions. If you can send more questions to my email. So yeah, please feel free to ask to us.
All right. Well, thank you again, Shige and Kazu, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Oriciro Genomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.